All right, finishing up with our gram-positive bacteria. Again, we're in our gram-positive, and we're in the bacillus shape, and we are in the bacteria that do not make endospores. So again, on that little flow chart, far right-hand side near the bottom. The next group or the next genus of bacteria found in this non-endospore gram-positive bacilli shape or gram-positive rod are the Mycoplasmas. Now, the Mycoplasmas, and I'm like, are the smallest free-living microbes, which means they don't always need a host to grow. They can grow and thrive out in the environment. Now, kind of a unique thing about this particular bacteria, it is a gram-positive, but it doesn't actually make peptidoglycan. And so if we gram stain it, it would show up as gram-negative, but genetically it is gram-positive. It doesn't have that double membrane that gram-negatives do. It does not have any of that lipid A. It would look gram-negative, but it actually shows up as, or it would look gram-negative, but it's genetically gram-positive. Also because it doesn't have that nice thick peptidoglycan that can help retain its shape, it's usually pliomorphic or pleomorphic, which means the shapes can vary amongst them. Also, when we try to grow these in lab, because we can, they start to get a little picky. They usually require various growth factors, either from a host or from some type of lab media. So they're usually a little more picky for what they need to grow. Places we can find mycoplasmas, they usually like to grow and thrive in the mucous membranes of both the respiratory tract and the urinary tract. And based on where they can grow, this is where they cause problems. And if they're in the respiratory tract, they can cause pneumonia. And if they're in the urinary tract, they can cause urinary tract infections. Only a few in the Mycoplasmas group really cause any type of significant disease. Like most of them are usually normal flora, or they cause kind of a minor disease, not all that deadly. Now, the one mycoplasma that does seem to cause the most problems that most people are familiar with is the Mycoplasma pneumonia. Again, it likes to live in the respiratory tract, and it causes what we call as atypical pneumonia. Also, more nickname that people may have more heard of it is walking pneumonia. Now, because it's atypical, it does not behave like normal pneumonia. The atypical symptoms is, that makes it atypical, there's no white blood cell response. Normally, if you have a bacterial infection, you have an increase in the number of white blood cells trying to get rid of the bacteria. It is unlike this particular bacteria. It does not cause an increase in white blood cell response. Also, it's called walking pneumonia because it is a less severe type of pneumonia symptoms, which means you can still be out and about, walking around. You're not hospitalized. It is usually spread by nasal secretions, and children the ages of between 5 and 15 are the most commonly diagnosed, mostly because they're not always all that clean. They don't wash their hands all the time, especially between blowing their noses. However, this can be found in adults as well. It's just more common in children and adolescent. Now, diagnosis, also it's atypical, is a little harder, mostly because it takes forever for these things to grow in a lab. When we grow things on Petri plates up in our lab, we pull them out in 24 hours and we have lots and lots of growth. For us to grow these on some type of media or some type of agar in our lab, it could take weeks, not just 24 hours, for the bacteria to actually start to grow up and for us to see the colonies. Our better diagnosis is usually some type of an ELISA that can specifically look for the antigens of this bacteria. Now, your best bet is just to not get it in the first place, and good hygiene is the best way, because again, it can be spread through those nasal secretions. Another bacteria that's gram-positive rods, with no endospores, the Corynebacterium. So when we gram stain it, yes, it gram stains positive. It's also pleomorphic, non-endosporing, so they're not all the same size of rods. So some are short little rods, some are longer rods. Some also like to group together in V shapes, some like to stack up in a palisade shape. A lot of these are non-pathogenic, normal skin, normal oral flora. Some of you, when we did throat swabs, actually ID'd out to a Corynebacterium. And I'm like, lots and lots of normal flora in this genus. However, again, they're not all normal flora. One pathogenic bacteria in this genus is Corynebacterium diphtheriae, which causes diphtheria. Now, one of the big symptoms of this bacteria is that this bacteria does create a toxin, and and as it starts, which blocks enzymes from working, which usually is going to cause some type of cell death somewhere. But as it does this, it actually can form cell death and a congregation of cells in the back of the throat. It's called a pseudomembrane. So you get these large growths at the back of the throat. Now, they can actually grow large enough that they can block the entire throat itself, actually causing death by suffocation. But it's a lot of fluid, white blood cells, as well as some clotting factors that form this big pseudomembrane. So it's also a diagnostic tool of seeing this as well. Now, so diagnosing this bacteria is, yes, whether you see the pseudomembrane or not, but otherwise to diagnose it, we're not always going to rely on the presence of a pseudomembrane. There can, one, grow it up on a type of specialized media called Loeffler's media. It's just a very specific type of agar. When we had all those different agars sitting out in lab, again, this is a very specialized media that grows specifically this bacteria. Otherwise, we can do various types of ELISAs as well. Treatment, they're going to give you an antitoxin or an antibody that's going to neutralize the toxin this bacteria makes. They can give you basic penicillin or erythromycin, and if you have a large pseudomembrane, a lot of times they'll do surgery to remove that so you don't have any blockage for breathing. Your best bet is to not get this in the first place, and we have a vaccine for it. It's the diphtheria toxoid vaccine. It is the D in DTaP. Since we have developed the vaccine, we have only had 12 cases of diphtheria reported in the United States in the last 15 years, so that's less than one case a year. Prior to the vaccine, we had hundreds of thousands of cases every year. So we go from 100,000 plus cases a year to less than one case a year, all because we now have the vaccine. So there's less people spreading it, there's less people that are getting it. And another group of bacteria, again, gram-positive rods, is our Mycobacterium. It's our last group. When we gram stain them, again, they're gram-positive, non-endospore. They're the rod-shaped. And the interesting thing about this group of bacteria in this genus is that this is the group of bacteria that has that mycolic acid. So the mycolic acid and the Mycobacterium, these are the ones that is that waxy lipid found in that cell wall. And so when we can't just gram stain these normally, yes, they would gram stain positive, but and I'm like, a more accurate stain is going to be that acid-fast stain. And I'm like, so genetically they're gram positive, but the stains won't stick. And I'm like, we have to do that acid-fast stain that has that acid alcohol. And we're looking for pink for our Mycobacterium. Now, unique characteristics of bacteria in this group. One, they are really slow to grow. It's not just 24 hours, it might be a week or more. Partly it just takes a little bit longer to make all that mycolic acid in their cell walls. They have a little more protection from breakdown after phagocytosis, so a lot of these are intracellular organisms that if they get eaten, they don't get broken down. And a lot of them are resistant to antimicrobial drugs. They're resistant to gram staining because of that mycolic acid. They're also resistant to heat and drying, chemicals, and various disinfectants. So having the mycolic acid makes them a little harder to kill as well. Now, there are 3 main bacteria that affect humans in the Mycobacterium group. There are lots and lots of Mycobacterium that are normal flora or non-pathogenic, but there are 3 that affect humans. And the first one is the Mycobacterium tuberculosis. Causes tuberculosis, also known as TB. Now, there are various types of, or progressions of tuberculosis. It is a pulmonary disease, as most people know. Luckily, our cases are decreased in the United States, but a lot of people are still harboring this bacteria, and it goes undiagnosed. The 3 types of tuberculosis really just has to do with the different stages of tuberculosis. The very first stage, and I'll go back and forth between this and the next slide, is called primary tuberculosis. This is that first initial infection. And so the bacteria just is getting into the lungs. And again, a lot of these are intracellular. So here's the bacteria. It's getting ingested by a macrophage, a big eater cell, but it does not get broken down. And so it's just getting inside the lungs. The second type is that secondary or reactivated tuberculosis. It's at this stage, and I'm like, when we start to have, well, let me go back to the primary tuberculosis. It's getting in the lungs, it's establishing, and as it establishes, all these infected macrophages stick together in what we call a tubercle. It's just a big, huge chunk of white blood cells that are all infected. That's where it gets its name, tuberculosis. Now, what can happen with this tuberculosis, or this tubercle, is after a while, these cells start to die, and they don't live forever. And so that's the caseous necrosis. It's these white blood cells dying. And what happens in secondary or reactivated tuberculosis, this tubercle ruptures, and now you have this bacteria getting released to go infect more parts of the lung. And so I'm like, it's just a rupture of the tubercle. Now, my little note, when this tubercle and these white blood cells die inside this tubercle, as this tubercle starts to break down, these dying cells actually create almost little like pockets. They're not really air pockets, but they appear that way. And as the cells start to break down, it almost starts to give it a yellowish look. And so they say there's a reason why this is holding cheese. They say this actually starts to look almost like a cheese-like appearance to it, where it has these holes as it's breaking down. Now, disseminated, excuse me for the coughing. The disseminated tuberculosis, so secondary, it's just rupturing and spreading. Go back. Disseminated tuberculosis is the bacteria is not just spreading inside the lungs anymore. It is now spreading to other parts of the body. This is showing some of those white cheese-like tubercles that are breaking down. You should never have that in your lungs. But disseminated is when it's now traveling to other parts of the body. It can get in the bloodstream, and it can travel to the spleen, the kidneys, the brain. It's a common bacteria found in a lot of immunocompromised patients, especially those that have HIV, that have almost no immune system. Now, diagnosing it, and I'm like, one, they can do an acid-fast smear. They can take a sample of, like a sputum sample, and do an acid-fast test on it. They can also try to grow the bacteria in the lab and look to see the colonies. They can look to do more further testing on it. They can do genetic probes looking specifically for this bacterium's DNA. They can do its PCR. They can also do the tuberculin skin test. And I'm like, that's where they're going to look for individuals that have been exposed to tuberculosis. They're going to inject the antigens for tuberculosis, not the whole living bacteria, but the antigens for tuberculosis into the skin. And if you have already made antibodies for this bacteria, those antibodies are going to react with those antigens that were injected, and you're going to form this big red bump. I'm guessing for most of you that have had the TB injection, the skin test, it did not look like that when they checked it the next day. But it's just looking for individuals who have been exposed to tuberculosis. It doesn't mean you have an active tuberculosis infection. And I'm like, but you have been exposed to it in the past, so you have antibodies. They can also do chest x-rays, and they're looking for these big tubercles inside the lung. Now, treatment is a little hard. A lot of common antimicrobials are not effective. And so they're usually going to try to do combinations of antibiotics. And even those, this is a hard bacteria to treat. It doesn't always act like others. It has that mycolic acid. It's just harder to treat. And so you're usually on some type of combination of antibiotics for 6 months, if not longer, to really try to get rid of all of these bacteria. These are some of the most common drugs that are given in their combination therapy. Your best bet is to get a vaccine. We've now developed a vaccine. It's called the BCG. It's named for the people that developed it. And wherever tuberculosis is prevalent, they can vaccinate you. Otherwise, it's being really careful around patients. Try not to inhale any respiratory droplets so that you're not picking it up. A second mycobacterium that can infect humans is the Mycobacterium leprae, which causes leprosy. It's found in cooler regions of the bodies, aka the extremities. It again still a mycobacterium. It still has the mycolic acid. But this particular bacterium grows best anywhere from about 30 to 34 degrees Celsius, so cooler than body temperature. So it likes those extremities. Again, it can also be grown in armadillos because they have a cooler body temperature. It's a rare bacteria to get. Generally, people get it. It's not extinct. It's still around. They can get it from person to person contact, usually with a break in skin or from nasal secretions. And I'm like exposed to some type of open wound or break in skin. It's also hard to treat because it's still a mycobacterium, and so it's usually treated like tuberculosis, combination of antibiotics for many months. And then our last mycobacterium bacteria is Mycobacterium avium-intracellulare, or also usually just shortened to Mycobacterium avium complex. It's the most common mycobacterium infection in AIDS patients, more common than tuberculosis. Individuals or patients get it from ingesting contaminated food or water that has this bacteria in it. But once it gets in the body, this particular bacterium, it's intracellular, it's all of this pink. And I'm like, it can get into any cell in the body. It's not just going to get into macrophages and likes to hang out in the lungs. It can go anywhere in the body, and it can get into just about any cell, which is going to lead to full organ failure. And it's extremely difficult to treat. So it is a deadly bacteria, especially for those that have AIDS. So it can affect anywhere in the body, and a lot of times it's trial and error for the antibiotics. What seems to be working, what seems to not be working, usually a combination, and it's usually a long-term treatment of this particular bacteria. And that's the end of our gram-positive. So make sure you take the online quiz, and I will see you, and make sure you watch the gram-negative intro, and I will see you in class.